Hi everyone, my name is Victoria Winter and this is my next presentation for my biology research on zebrafish and embryology. The article that I chose for today's presentation is called Drinks Like a Fish, The Effects of Ethanol on Associative Learning in Zebrafish. And it's by the authors Fort Negley and Nick Owen. I really wanted to choose this article because it says a lot about um, like how ethanol affects zebrafish cognitively and with their associative learning, which I feel like relates to autism. I work a lot with autistic kids, and a lot of them have been affected by substance abuse disorders from their parents, um, and their mom drinking alcohol or doing drugs whenever she's pregnant with them. So this paper in particular seemed really interesting to me. So let's go ahead and get started. So this study sought to see the way ethanol impairs young zebrafish cognitively. The scientists conditioned 3-month-old zebrafish to find their ways through a maze. They exposed the zebrafish to different amounts of ethanol and tested the way that different concentrations affected cognitive remembrance. They wanted to use older zebrafish rather than just regular embryos like what we're using in our research, because they wanted to test if further neurological maturation can better protect against the teratogenic effects of ethanol on development and also remembering and learning. They also determined the times it took for the fish to complete the maze and compared the times and concentrations to determine which concentration and time had the most detrimental effect on the way the fish remembered their way throughout the maze whenever they were conditioned to find it before. So the main hypothesis presented in this article was that further neural maturation can combat the negative effects of alcohol on the brain in zebrafish, which in turn will coincide with humans because of the genetic similarity we have to zebrafish. This means that the more a pathway is developed in the brain, the less likely that alcohol will affect this pathway. For example, the better a fish memorizes its way through the maze, the less the alcohol will negatively impair the fish's abilities to find its way through when it has been exposed to alcohol. So the most notable finding of this study was the significant time difference between the control group and the exposure group. This graph shows that fish that were exposed to ethanol took a significantly longer time to complete the maze than the fish that were not exposed to ethanol. As you can see, the data spikes up drastically at 0.5 days, but then slowly creeps down after that. This is indicative of recovery after exposure to the ethanol when the fish metabolize it and it no longer affects them as strongly. This shows that recovery from exposure to ethanol is possible in zebrafish embryos and also possible in humans in turn through implications. So, significant differences between the way that embryos and older zebrafish were found, strengthening the hypothesis suggesting that further neural development protects against the effects of ethanol on zebrafish. The 3-month-old zebrafish are able to metabolize ethanol better than embryos, most likely because their um, brain is more developed and they have better and stronger neural pathways, and also possibly because their liver is more developed. But that wasn't mentioned in the paper. Um, it's just my remembrance. So what the scientists also found was that the data that the data points to the possibility of recovery from ethanol in these zebrafish. It shows that although that the fish were exposed and negatively affected for some time by the ethanol, they were able to metabolize the ethanol and overcome the effects of the alcohol over time. It was not overcome completely, but partial recovery was still observed. This study also pointed to evidence that zebrafish have working memories like humans do. They can learn information and procedures like going through a maze. And this is really groundbreaking because it's similar to the way that humans act. Humans can learn things and have associative memory, and we also see in our lives that alcohol can negatively affect these pathways in humans. So it's great to see that it also affects zebrafish in the same way. So this research shows that ethanol has a negative effect on associative memory shortly after zebrafish are exposed to it. It also shows that the older a zebrafish is, the better it metabolizes the alcohol. It shows possibility for recovery post-exposure, which is great for zebrafish because it's similar to humans. This has implications for humans because of the genetic similarity between zebrafish and humans. Humans metabolize alcohol in a similar way to zebrafish. This can help with fetal alcohol syndrome research in humans because it shows that the later on in neurological development ethanol exposure occurs, the better outcomes occur. So the information presented in this article is extremely helpful for my research project. It gives a lot of background information, which is helpful when learning about ethanol toxicity in zebrafish, especially in a model that relates to humans. This article tells the cognitive similarities between zebrafish and humans and how ethanol negatively impacts both. This research project also mentions ways in which zebrafish were exposed to ethanol concentrations, which will be helpful for my project. It gives me ideas on how to test for different behaviors present because of the altered neurological function in the fish because of the ethanol exposures. Neurological function greatly interests me because of my work with autistic children. I would love to incorporate a behavioral study like this one in my research to test the way fetal alcohol syndrome can cause behavioral defects like autism and other cognitive and neurological defects. So thank you all for listening. Here is the citation for the article. I really appreciate it.